Good afternoon. My name is Sophie Jensen, and I want to tell you about a new wave of flavors. As you might have noticed, the utilization of seaweed has become increasingly popular. Even though quite a few edible seaweed species grow naturally along the shores of Western world countries, this natural resource is currently not being used to its full potential within the food industry, as there are few commercially available food products that include seaweed on the market today. Since they are abundant in minerals, seaweeds have a naturally salty taste which can improve the flavor profile of foodstuffs. The flavoring properties of seaweeds are, however, still quite unexplored, and the development of seaweed food products still relies on more research efforts. So here I will present our work at Matís to develop flavor ingredients from edible seaweeds. The work included enzymatic processing of Ascophyllum nodosum and Saccharina latissima to produce flavor compounds with sodium replacing and flavor enhancing properties. Seaweeds does not only contain minerals, but also a range of potential flavor components that can enhance the flavor of food. In particular, the proteinaceous compounds can be responsible for enhancing flavor characteristics such as umami, meaty, and roasted flavor, in addition to providing textural mouthfeel. The problem here, though, is that the cell walls of brown algae are mostly made of alginic acid and cellulose. So to unlock these flavor components that impart umami flavor, polysaccharide degrading enzymes such as alginate lyase enzymes are needed to help break down the algal cell wall. In this project, we used two enzymes in the processing of the seaweeds. One was a commercial umamizyme purchased from Amano Enzyme Europe Limited, and the other one was an in-house alginate lyase, ALK3, which was developed and produced at Matís. ALK3 is a thermostable enzyme originating from Rhodothermus marinus that catalyzes depolymerization of alginate by beta elimination of the beta-1,4 glycosidic bonding. ALK3 is an endotype enzyme that is active on both manuronic and glucuronic blocks of alginate. Bacillus subtilis was used as the production host for ALK3, which were prepared and the plasmid DNA transformed according to the Groningen method. ALK3 enzyme was then extracted from the cells by means of sonication, heat purification, and centrifugation. The wild biomass of the two seaweed species, Saccharina latissima and Ascophyllum nodosum, which are both indigenous in Iceland, were collected in Breiðafjörður in Iceland. The bulk seaweed biomass collected included blades, stipes, and holdfasts. The biomass was wet milled, freeze-dried, and homogenized before the enzyme degradation. We produced control samples without applying any enzymes, and reference samples were extracted applying only one enzyme, so either ALK3 or umamizyme. And for the purpose of upscaling, we tested different units of ALK3 with different degradation durations. The produced seaweed extracts were then evaluated for their chemical as well as sensory properties, and then applied in different food models to evaluate the potential of reducing the sodium content while sustaining umami flavor and flavor richness. Sodium content was determined both in extracts as well as the raw material. No significant difference in sodium content was found in the different seaweed extracts, but as you can see here, the sodium content is significantly reduced in the extract during the processing. The total polyphenol content was determined, but no difference could be found between the extracts, and it was not affected by the processing. Polyphenols are known to have a bitter taste, so a reduction in the TPC would have been a positive effect on reducing the bitter taste of the extracts. Extracts were also analyzed for monosaccharide content according to the NRL determination of total carbohydrates in algal biomass. And the main purpose of the monosaccharide analysis was to determine the amount of rhamnose. Since rhamnose can, depending on linkage position to other moieties and attached groups, induce bitterness, which may affect the possible use of the product as a flavoring agent. Rhamnose was under the limit of quantification or limit of detection in all seaweed extract samples analyzed. So it was concluded that rhamnose is unlikely to contribute to any perceived bitterness of the seaweed extracts. The odor of the produced seaweed extracts were evaluated by 3 trained sensory judges, and their compiled opinions are shown here in this table. According to the results, extracts 3 and 7 have a meatier odor resembling that of a meat broth cube. And the results from this trial served as a basis for continued work using both ALG3 and umamizyme in the extraction procedure. The taste profile of 8 seaweed extracts were evaluated by electronic tongue for acidity, salty, bitterness, umami, astringency, and aftertaste. The e-tongue characterizes the taste profile of samples based on the selective attraction of different taste molecules through sensors that consist of unique lipid membrane. The taste molecules bind according to electrical and hydrophobic attractions, and an electric signal is obtained by a potentiometer sensor compared to a reference electrode without the membrane. According to the results here in the PCA plot, there's a clear distinction between the samples treated with both ALG3 and umamizyme, so that's samples 3, 4, 5, and 7, and the control samples, samples 1 and 2, as well as samples treated with only one enzyme, samples 8 and 9. Sample 3 and 5 are also close to each other, confirming that 5 or 50 units of enzyme per gram of raw saccharina material gives a similar extract outcome. These results were used as a basis for a scale-up of the enzymatic processing. Two seaweed extracts were assessed for stimulation of ATP in bitter-responsive human taste bud epithelial cells. The principle here is that the receptors for bitter taste, they will release calcium when triggered by a bitter taste agonist. Intracellular calcium ions released by activation of the receptors will stimulate ATP secretion. ATP acts as a neurotransmitter to activate sensory nerves, and the luciferase-luciferin reaction allows detection of ATP as proportional luminescence. The estimated IC50 for eliciting a bitter taste response in individual as well as pooled donor-derived bitter-responsive HTBEC for the two seaweed extracts was found to be 1 milligram, approximately 1 milligram per milliliter, or 0.1%, which seems very low, but this test is very sensitive. We then produced selected seafood models to compare salt versus seaweed extracts as salt reducers and/or flavor enhancers. The level of salt reduction achieved with addition of seaweed flavor powder was assessed by trained sensory panels. This sensory-guided development will give us valuable information of the usability and market of the flavor ingredients. The results from the sensory trials of the different food models indicate that these seaweed extracts would not be a suitable ingredient in mashed potatoes due to off flavor and odor. In cooked poultry products marinated with Saccharina latissima extract at 1.6%, umami flavor was detected, although its intensity was lower than MSG and the commercial umami flavor. Ascophyllum nodosum samples provided a high-impact marine flavor, while marine flavor was not detected in the Saccharina latissima samples. But this needs to be studied further. Fish cakes containing extracts from Ascophyllum nodosum were characterized by off-odor and flavor, and a weaker fish cake odor and flavor compared to traditional fish cakes. Salty taste was strongest in the fish cake control samples containing 1% salt, and in the fish cakes with 30% sodium reduction containing extract from Saccharina latissima. So in this study, extracts of two seaweed species, Ascophyllum nodosum and Saccharina latissima, were successfully produced with enzymatic hydrolysis, applying an in-house produced alginase, ALG3, and a commercial umamizyme. The extraction process was modified for upscaling, and extracts could be produced in sufficient amounts to enable their implementation into different food models and be evaluated in live sensory panels. The results obtained flavor components that are rich in umami and salty taste, with the potential to replace sodium and create flavor-enhancing ingredients for food products. Future research focusing on further development and adaptation to scale up of the enzyme production and food application development is envisaged. Furthermore, a feasibility assessment is needed to, for example, study if the process is economically beneficial. Thank you very much for your attention.